recording. So we want to welcome you to the second year of our virtual Center for Iron and Heme Disorders seminar series. Want to welcome those who are new to join us and those who have come back. This is a really fun series for us to be able to learn state-of-the-art science that's going on in the iron and heme community, and also establish those collaborations and learn more about what's available for us at the different cores. So I'm going to share my screen right now. See if I can get to that, share it, this one right there. So I just am referring you to the website for the Center for Iron and Heme Disorders, where you should be able to see several different things here. One of which is the three key cores that we have at the Center for Iron and Heme Disorders. There's an iron and heme core that can measure metals for you by ICP-MS, as well as measure the biosynthetic enzymes and products in the heme biosynthesis pathway. There's a mutation generation detection core that can help you with state-of-the-art mutation tools, such as TALENs and CRISPRs. And also in designing those and also generating lentiviral systems to make mutations in your system. And finally, there's a metabolomics core, which can do metabolites, lipidomics, as well as quantification of those molecules. And I highly recommend you go and read about those resources. This is one of the U54s. There is also we also manage a greater coordination center, which is the coordinating center for all the U54s. And it's this website here called the CCEH.io. This is the coordinating center for the different cores around the country. And there are facilities at several different locations, including Yale, at Harvard, at the Fred Hutch, at Indiana, and here at Utah. So I highly recommend you go to these sites and look at things. And one of the things I want you to specifically look at is this funding page, where we have a new call out for P&F funds. There are two types that are available. One are the type A grants, which are $5,000 to $12,000 worth of what we call core credits, which is credit at any core at any of the centers that I just talked about, where you can bank credit for a project that you are interested in utilizing cores that are sponsored by the NIDDK hematology projects. The second type of grants are these type B grants right here, and they're up to $75,000. They're specifically focused on individuals who are up and coming and new, so postdocs, new faculty members, and these grants are up to $75,000. A core use is not required, but it's encouraged. And the new call for applications is due August 1st. And you can read through the details of this here. So I recommend highly that you go investigate those websites and the cores that are available to improve and enhance your research in hematology. So now I want to introduce Shang. So Shang comes to us. He grew up in China, and then for his bachelor's degree went to the University of British Columbia in Vancouver, where he got his degree in genetics. He then moved on to the University of Wisconsin-Madison and got his Ph.D. in cellular and molecular biology in the laboratory of Dr. Zhen Huang. Where he actually was very prolific and published and focused on development, neural development, and the communication between the vasculature and the neural development, and identified several signaling molecules that are important in that development. He then went on to the laboratory. I should have asked this question, and I'm sorry. Went on to the laboratory of Ardem Patapoutian.
Patapoutian.
Thank you. I'm so sorry. I should have asked that before.
No problem.
Where he's been working on understanding the molecule piezo and in red cell dehydration, but also recently, and his subject of his talk today is the role of piezo in iron metabolism in mice and humans. And We want to thank Shang for talking. And just for those of you who are attending, Shang is on the job market. And so if you have any interest in contacting him, this is a great opportunity to learn about his research. So I'm going to let you go ahead and share your screen now, Shang, and you'll have about 45 minutes. If you have questions, please populate them in the chat, and then I can monitor them, and I will either ask the question or invite you to unmute and ask your question. And Shang has agreed it's more fun to have interruptions and talk about things as we're going on. So please feel free to do that.
Okay, so yeah, thank you very much. That is a really, really nice introduction. Okay, so now I'm sharing the screen.
Yep.
I think you can hear.
Looks good.
And you can see. Okay, yeah, let's get started. So hello, everyone. So my name is Shang. I'm a postdoc from Patapoutian's group from the Scripps Research Institute. So today, the title of my talk is the role of piezo-1 in iron metabolism in mice and humans. So,
sorry to interrupt. It looks like it's presenting as your PowerPoint and not a...
Oh, okay, I see. Okay, then I play with this full screen.
Perfect.
Yeah, okay, thank you. Okay, thanks. So yeah, so first of all, I want to thank for inviting me to this seminar series. So this is a very exciting opportunity to talk to the expert in the iron field. So today, actually, instead of focusing on the iron metabolism, I'm going to do something different. That I'm going to tell you a story about how we get started with the study of a very important and interesting ion channel, and how our research unexpectedly came into the field of iron metabolism. So as you probably know, that Patapoutian's lab focused on the field of mechanotransduction. So in cell biology, mechanotransduction is a conversion of mechanical force into biological signals. And this can be achieved by activation of ion channels, because ion channels are perfect molecular switch for the cells, because they can be open very quickly in response to external stimuli, such as chemicals and the voltage. And then the influx ion can get into the cell to activate downstream signalings. However, the mechanically activated ion channels had not been known in mammals for a long time. About 10 years ago, Patapoutian's group discovered a novel family of mechanically activated ion channel called piezo. So basically, they performed RNA screen in mammalian mechanosensitive cells and identified piezo-1 and the piezo-2. So as you can see from here, piezo channel is pretty large, which has 38 transmemory domains. That is more than any other protein in human genomes. And from the QuEM structure, you can see that the piezo proteins come together at the trimer and spread over on the plasma memory. And it is so large that it is predicted that the whole structure can bend over the plasma memory. Okay. So importantly, piezos are first bona fide mammalian mechanically activated ion channels. So that means they are sufficient to give rise mechanosensitivity to the mechanically insensitive cells. And
Shang,
the piezos, okay?
I need to interrupt one more time.
Sure.
You're a little quiet.
Okay.
I can hear you, but it's just a little quiet. Thank you.
So yeah. Okay, and also piezos are highly conserved in both animals and the plants, suggesting that they have important evolutionary functions. And more importantly, piezos are professional mechanosensors, because these ion channels, they only do one thing and one thing only, that is to detect physical force from the plasma memory. So after the discovery of piezo ion channels, Patapoutian's groups, together with other groups from the world, study the function of these important molecules. So in general, piezo-2, so in mammalian cells, there are two piezos, piezo-1 and piezo-2. So in general, piezo-2 expressed in sensory neurons are the mechanosensors for detecting mechanical force from touch and the muscle movement, as well as mechanical forces generated from internal organs like the lung and the bladders. On the other hand, piezo-1 is important in mechanotransduction in non-neuronal tissues, such as blood vessel cells, as well as bone cells. However, so in principle, most cell types in vivo can experience physical force, either in their static state or during motion. So mechanotransduction is expected to have a broader significance in biology. And one major challenge of this field is to identify and study underexplored mechanotransduction in vivo. And the question becomes how we are going to identify these novel mechanotransduction processes. So to solve these problems, in Patapoutian's lab, I have been taking a unique approach of combining human and the mouse genetics approach. So what I did is that I identified interesting human disease phenotypes from patients that carry rare piezo mutations in their genomes. And then I used the new animal models to study the underlying mechanisms. So one of the disorder I studied is xerocytosis, which is characterized by red blood cell dehydration. So as you can see from the picture that the red blood cells from the xerocytosis patients are dehydrated and have abnormal morphology compared to the normal blood. And the xerocytosis can lead to mild anemia and splenomegaly, but sometimes this disorder is asymptomatic. So it is considered as a very rare disorder because so far there are only 120 cases in a few Caucasian families caused by 20 different gain-of-function piezo-1 mutations. So these disease-causing mutations are gain-of-function because the mutated channels have larger inactivation time. That simply means these mutated channels can be activated normally just as wild-type copies. Wild-type channels, but after activation, the mutated channels are inactivated much slower than the wild-type, and this allows more ions, including calcium, get into the cell to cause overactive downstream signaling pathways. So that's why they are defined as gain-of-function mutations. So to study the mechanisms of piezo-1 dependent xerocytosis, we engineered a tissue-specific gain-of-function piezo-1 animal model. So we are very lucky to be able to do it because one particular gain-of-function mutation is located in the last exon of the piezo-1 gene. So in the mouse genome, we just fluxed the wild-type copy, wild-type last exons. And then immediately downstream of it, we engineered a new same last exons, but has the gain-of-function mutation in it. So in the presence of Cre recombinase, the wild-type copies are removed and the gene only express the cDNA that has the gain-of-function mutation in the genome. So to mimic the human disorder situation, I first created the constitutive gain-of-function mice, which means the gain-of-function piezo-1 allele is induced in every cell types in this animal. And I observed that the red blood cells from these gain-of-function animals are dehydrated and have abnormal morphology compared to the wild type. And later on, we showed that when we induce this gain-of-function mutation only in the red blood cells, it fully recapitulates these defects. So suggesting that our model can be a good animal model for studying the mechanism of this particular disorder. And with additional in vivo and the in vitro experiment, we learned something new about red blood cell mechanotransduction dependent on piezo-1. So I put a summary here. So in general, so when the red blood cells navigate in the circulation, they need to actively adjust their cell volumes because they need to be squeezed into smaller spaces. And in this process, there is a lot of squeezing mechanical force applied on the red blood cells. And in the experiment, we showed that the piezo-1 is a mechanosensor for this process. And at the molecular level, what happened is that the piezo-1 ion channel can be activated by the mechanical force in the circulation. And this leads to the increase of intracellular calcium. And that results in the loss of water and the potassium ion from the red blood cells. And then in this way, the red blood cells become smaller. So that's how the red blood cells adjust their volumes in response to mechanical force in the circulation. However, in the case of their cytosis patients, the whole pathway becomes overactive because of the gain of function PSO1 mutation. So that leads to the excessive loss of water from the red blood cells, leading to red blood cell dehydration. So that's how we explain the phenotype caused by the gain of function PSO1 on the red blood cells. And importantly, by a loss of function analysis, we showed that the PSO1 is required for red blood cell volume regulation, because if we delete piezo from the red blood cells becomes bigger or overhydrated. So that suggests that this important ion channel mechanosensor have important function in red blood cell physiology. And also, in addition to the study of the cell cytosis disease mechanism, I also use this animal model to explore a very unexpected and interesting relationship between infectious disease and the red blood cell disorder. Because from a very interesting old literature, we know that the cell cytosis dehydrated red blood cells are highly resistant to the infection of Plasmodium, which cause the human malaria. So since the PSO1 gain of function also causes the red blood cell dehydration, we want to know whether this is true in vivo. So whether the PSO1 gain of function can also induce resistance to malaria. I think the gain of function model is great a tool to explore this. So
there is one question in the chat.
Okay.
So if you go back 2 slides, yeah, the question populated is the problem a volume loss or is it the potassium loss itself? Or are they, concomitant? They go together. So you can't tease it apart.
So actually, they actually almost happened together. So we actually didn't tease them apart, whether it's because of the potential, potassium of the water. But I think that if you lose water from the red blood cells, they become dehydrated and the cell volume becomes smaller. So that is our working hypothesis.
Thanks.
Yeah, thank you. So, and indeed, when we infected the animal, the wild-type animal with the rodent-specific Plasmodium, actually the wild-type animal died between 5 to 10 days. However, the gain-of-function mice, either heterozygote or the homozygotes, can survive significantly longer after malaria infection than the wild-type, suggesting that the gain-of-function PSO1 actually confers resistance to malaria infection in vivo to the rodents. And remarkably, all these gain-of-function animals are completely protected against the cerebral malaria. So a situation where the infected red blood cells travel to the brain and cause the blood-brain barrier breakdown and associated problems. But as you can see, that the gain-of-function animals are completely protected. So from this malaria perspective, our work is quite similar to the story of sickle cell, because sickle cell is well known that the sickle cell disorders are highly resistant to infection of malaria. That's why we have a high prevalence of sickle cell disease allele in the African population where malaria is a major problem. So since the PSO1 gain of function allele also confer malaria resistance, we wondered whether the gain of function PSO1 allele actually are present at a high level in African population. So to answer this question, I did a functional screening for the gain of function PSO1 allele in African population. So first of all, I performed the data mining into different human genomics database, and I identified and cloned all the missense PSO1 mutations that are only prevalent in African population. And then I expressed those those PSO1 mutant in the hex cells, and then I used the UDO1 induced calcium signaling at the readout to identify potentially gain of function PSO1 mutation. So the UDO1 is a small molecule that can specifically activate the piezo1 and cause the influx of calcium into the cell. So by using this setup, I identified the two piezo1 mutations. When overexpressed in HEC cells, they give rise to larger calcium signaling in response to UDO1 activation. So suggesting that these two mutations might be the gain of function piezo1 mutation. And indeed, our electrophysiology confirmed this result, that we showed that these two mutated channels actually have much larger in activation type, but the effect is still smaller than the strongest rare gain of function mutation isolated from those rare cell cytosis patients, suggesting that these two mutations are mild gain of function mutations. And amazingly, one of these mutations, E756 delete, which delete a single amino acid from the N-terminals of the protein, has a very high allele frequency only in the African population, but not in other populations. So this result itself is very surprising, because when translating into the genotype frequency, that means 1/3 of major African populations, including African Americans, at least carry one copy of this allele in their genomes. So that means the gain of function piezo1 mutation not only exists in African population, but it exists in very high level. And we propose that this is because the gain of function piezo1 mutation confers significant and important resistance to malaria. To test this hypothesis experimentally, I obtained red blood cell samples from African American individuals who carry this gain of function allele in their genomes. And what I observed is quite surprising and significant, that some of their red blood cells show this dehydrated morphology compared to the non-carrier wild-type African American individuals. But we should notice that most of the red blood cells are still normal. So that is expected, because the gain of function mutation we identified here is only a mild gain of function activity. But more importantly, when we infected these red blood cells with the plasmodium in vitro, these dehydrated red blood cells have a reduced level of infection compared to the wild type. So this is consistent with our hypothesis. And importantly, last year, there's an independent report that confirmed our result with epidemiology evidence, in which they suggest that the E756 delete gain of function allele is associated with less severe malaria in a cohort of African children. So which provides important support for our central hypothesis.
Question?
Yes.
Is the absolute parasitemia reduced because the parasite can't get into the red cell, or they can't proliferate?
That's very interesting question. Actually, at this moment, we haven't figured it out. So, but this is going to be a next interesting question that we are going to try to answer. So I hope we can use some like a high resolution imaging approach to actually observe what happens at cellular and molecular level during the infection. But that is an important question.
Thank you.
Yeah, thanks. So yeah, to summarize this part, actually, in addition to the importance of this, the importance to the malaria and the red blood cell disorder, the discovery of this very common gain of function allele in African population will allow us to have a deeper understanding of mechanotransduction from human genetics perspective. For example, at this moment, for any medical conditions observed in these rare cell cytosis patients, we can now use the gain of function animal model to study the underlying mechanism. And at the same time, we can evaluate whether the similar medical conditions affects these African American individuals who carry this gain of function allele in the genomes. So right now, so now I'm going to highlight a story from this perspective. So that brings us to the study of iron overload, because in addition to the red blood cell dehydration defects, so the xerocytosis patients with gain of function PAS1 mutation also develop late onset iron overload. So which is characterized by the excessive iron in the body that damages the multiple organs. So in general, iron overload is a rare disorder affecting only 0.5% of Caucasians with certain genetic mutations. And I think this iron overload is a medically underappreciated disorder because there are only a few options for the treatment, including chelator and the bleeding. However, in the case of xerocytosis patients, the mechanisms for causing iron overload in these patients is still not clear, although it is believed to be caused by red blood cell defects. So I think since we have this tissue-specific gain of function animal model, it is a great opportunity to test and study this mechanism in vivo. So first of all, our gain of function PAS1 mice also develop late onset iron overload because we observe this excessive iron accumulation in the livers of aging gain of function PAS1 mice, but not in the wild type. So the iron is stained by the blue color staining here. So, and this effect actually does not happen in the younger adults. So that's why it's a late onset. And in addition to the histology, I also measure the transferrin saturation value. So transferrin is a small protein in the blood that transports iron in the circulation. And the saturation is a percentage of the total iron binding capacity. So the higher value indicates excessive iron in the blood. And we observed a significant increase of transferrin saturation value in the aging gain of function animals. And this did not happen in the younger adults. And we also observed a similar result for the ferritin values in these animals. So the ferritin is also a small intracellular peptide that binds iron and stores it in the tissue. So it's a blood value reflects how much iron you have in your body. So based on this histology and the blood measurement experiments, our gain of function animal model are a great tool to actually study the iron overload caused by gain of function PAS1 mutation. So because of this, I decided to determine the cell type specific requirements of gain of function PAS1 in causing iron overload. So as you probably know better than me, the iron metabolism is a very complex and process that involves the multiple cell types and organs. So first of all, the enterocytes from the small intestine can absorb the iron from the food, but this only accounts for less than 10% of the total iron in the circulation. So the most majority of the iron in the circulation comes from the recycling of the senescence of red blood cells by macrophages. So basically, the macrophage phagocytose the old red blood cells and release the iron from the hemoglobin into the bloodstream. And the hepatocytes are also important for maintaining the iron homeostasis in vivo, because in response to the higher level of iron in the blood, the hepatocytes secretes small peptide hepcidin, which inhibits the mobilization of iron into the bloodstream. And importantly, the erythropoiesis are also a very strong contributor to the iron pathway, because in the case of overactive red blood cell recycling, the erythropoiesis becomes stressful because there's a higher demand for the iron to make new red blood cells. And in this case, the erythropoietin hormone is released from the bone marrows as well as spleen to actually inhibit the production of hepcidin. And the net result is to make sure there's enough level of iron is released into the bloodstream to make new red blood cells. So since PAS1 is highly expressed in the red blood cells as well as the macrophages, I then evaluated the contribution of these two cell types to the development of iron overload in our animal model.
I'm going to interrupt 1 second. John Phillips asked a question.
Sure.
John, do you want to go ahead or you want me to ask?
Sure. Is there a gender difference between iron overload that you see?
So you mean in our animal models?
Yeah. Mhm.
So actually, in our animal models, we actually didn't observe the gender difference in the iron effect, but actually we noticed the females have a bigger variability in terms of iron levels. So I think it may depend on the cycles.
Okay.
Yeah.
Thank you.
Thank you. Thank you very much. So now we came to the real genetic experiments with our animal models. So surprisingly, we found that the gain of function piezo1 in macrophages, but not in the red blood cells, cause iron overload, since we only observed the iron accumulation in the livers of macrophage-specific gain of function mice, but not in the red blood cell-specific gain of function animals. So this result actually challenged the conventional thinking that the iron problems are secondary to the red blood cell defects. So instead, it proposed that the macrophage mechanotransduction, which is dependent on piezo1, might be important in iron metabolism. And the question now is the mechanism. How does overactive piezo1 in macrophages cause iron overload in the animals? So as I introduced, since the hepcidin is a master regulator of iron pathway in vivo, I measure its level in our animal models. And first, I noticed a sharp decrease of hepcidin level in the aging macrophage-specific gain of function animals compared to the wild types. And since the hepcidin is a negative regulator of iron pathway, this is consistent with our iron overload phenotypes. And interestingly, even for the younger adults, when we didn't observe the iron overload phenotypes, there's a significant but mild decrease of hepcidin level in the macrophage-specific gain of function mice, suggesting that at the younger age, the hepcidin pathway is already compromised.
There's a question in
quick
the chat.
question.
Yeah. Yes.
Yeah, just a quick question. Did you
Yes.
also look at the macrophages of the spleen, or is this only the Kupffer cells of the liver?
Actually, when we do the analysis, we mainly analyze the macrophage from the spleen, because that's where the red blood cells are recycled by the macrophages. Yeah. But actually, when we did the histology, we also looked at the macrophage in the hepatocytes, and we observed that they are overloaded by the iron.
Cool.
Yeah. Thank you.
Thank you.
Yeah. So, and also there's this experiment on the younger adult also suggests that the younger adult, although they haven't developed the iron overload phenotype, they may have a compromise in the acute response to the iron stress. To test this hypothesis, I did an experiment in which I injected iron dextrin into the animals for 24 hours. So in this case, it's just a wild type animal. And then this treatment actually is not strong enough to alter the iron metabolism for the wild type animals. However, when I did the same experiment on the gain of function animal, we observe a significant increase in the blood iron after this response, after this treatment. And also we showed that this is specific to the macrophage specific gain of function animals, suggesting that the younger adults actually with compromised hepcidin in the gain of function mice are susceptible to perturbed iron stress, are more susceptible to the quick iron stress.
We do have one more question in the chat, which is
Yes.
Thomas Coates. Do you want to ask your question, Thomas?
Yeah, I'm just curious if you've looked in the heart of these animals,
Yeah.
for iron levels.
Yeah, thank you. So actually, we looked at the spleen, the heart, and the liver, and we found also in the heart and the spleen there's a excessive accumulation of the iron by the chemical staining. So that's actually these animals at the older age, they tend to actually die earlier than the wild type. So they, most animals, they can survive 2 years without any problem, but these animals, they probably died, after 1 or 1.5 years. So maybe it is because of iron overload in the critical organs, but we cannot actually, determine whether that is the case or not. But yes, we did. Yeah.
We could discuss, I don't want to take time here, but this happens in humans also.
Oh, I see.
One patients, yeah.
I see. Well, that's interesting. Yeah, thank you.
You can talk at the end.
Yeah. So, and also next, we found that the stressful erythropoiesis in the macrophage-specific gain of function PAS1 mice are actually also happened in these animal models. So as I introduced, the stressful erythropoiesis are important to actually suppress the production of hepcidin. And I looked at the level of this critical hormone. And what I found is that there is a sharp increase of erythropoietin in the macrophage-specific gain of function animals compared to the wild type. So usually in wild type, this hormone is almost undetectable. And this is consistent with the reduction in the reticulocyte number in these animals, suggesting that the erythropoiesis becomes stressful. And then I further proposed that this stressful erythropoiesis is caused by the increased red blood cell recycling as a result of enhanced phagocytosis of macrophages. And to test that, first I evaluated the red blood cell turnover in these animals. So I performed a well-established experiment in which I fluorescently labeled red blood cells in vitro and injected into the animals. And then I measure the percentage of the fluorescent red blood cells in the circulation from the animal. And then as expected, the number of fluorescently labeled red blood cells decline because they are phagocytosed by the macrophages. But what I observed is quite remarkable that in the gain of function PAS1 animals, the turnover rate of these fluorescently labeled red blood cells are significantly increased compared to the wild type, suggesting that the red blood cell turnover is increased in this animal. And here is a measurement for the red blood cell half-time, consistent with the decline rate of these fluorescent labeled red blood cells. And then I evaluated whether the PAS1 gain of function affect the phagocytosis. So as you can see from this video, the phagocytosis is very mechanically active process that involves a lot of plasma-membrane tension, especially at the site of engulfment. But at the molecular level, how the plasma-membrane mechanical force is sensed by the cell and how that translates into downstream signaling during phagocytosis is unknown. So here I hypothesize that the PAS1 as a mechanosensor is important in this process. So first, we measured the phagocytosis in the spleen. So we looked at the macrophage from the spleen, and by co-labeling approach, we determined that in the gain of function animal, there are significant higher level of internalized red blood cells in the spleen macrophages, suggesting that in vivo, the macrophages with the gain of function PAS1 phagocytose more red blood cells. And this is consistent with the in vitro data where we observed that the macrophage with the gain of function PAS1 mutation, they internalize more red blood cells compared to the wild type. So these two experiments suggest that the overactive PAS1 enhance the phagocytosis ability of macrophage.
Can I ask a quick question again? Are
Yes.
those wild type red blood cells or PAS1? It's just the
so which one you refer to?
for the purified red blood. What's your for the video?
For the video, yeah. So this is actually the red blood cells, and these are the bone marrow derived macrophages.
They're from the macrophage specific piezo mutation?
From wild type. This is just to show the the phagocytosis in real time.
Can I ask a follow-up question as well? What about
Yes.
the half-life study then, the previous?
For the half-time, actually we induced the gain of function piezo in all macrophages, not only in the spleen, but all the macrophages lineage.
One other question.
But the red cells, so the red cells are not gain of function then?
No, the red blood cells are well typed. This is only specific in the macrophage. Yeah, the red blood cells are not affected, are not expressing the gain of function piezo.
One other question in the chat. Would you like to unmute and ask it? Sound the cock. I hope I pronounced that right.
Yeah.
The question is, do you think sickle cells have increased recycling in the piezo?
The sickle cell. I see. So actually, this is an interesting question. We actually didn't perform any experiments on that. So I don't know.
Good, thank you.
Yeah, thanks.
Yeah, okay.
Okay. So then we try to get more mechanisms of how the piezo-1 can link mechanotransduction to phagocytosis. So it is well known that during phagocytosis, the plasma membrane tension plays an important role in this process. So there's a dynamics plasma membrane tension in this process. And at the molecular level, the calcium-dependent REC1 signaling is important because the REC1 as a small GTPase actually promotes the actin filaments dynamics in this process. So that's why our hypothesis is that the gain of function piezo-1 can elevate intracellular calcium level, which promote REC1 activity. And as a result, the phagocytosis is enhanced. So first, we show that in vitro that the macrophage expressing the gain of function piezo-1 protein can induce a larger amount of intracellular calcium in response to the UDA1, the agonist for the piezo. And then we also measure the REC1 activity in the gain of function 1 macrophages in vitro. And we show that during the phagocytosis process, the REC1 activity is increased compared to the well-typed macrophage, suggesting that piezo-1 gain of function, the overactive piezo-1 actually increased the REC1 activity in vitro. And finally, we want to ask whether piezo-1 actually is required for iron metabolism in vivo, and also whether it's required for phagocytosis. And to do this experiment, I actually injected the iron dextrin at higher level into the animal for 5 consecutive days. And this treatment was strong enough to elevate the iron blood measurement in the wild-type animal. But when we delete piezo-1 specifically from the macrophage, this response was completely blunted, suggesting the piezo-1 is required for acute response to the iron disruption. And consistently in vitro, we showed that the macrophage without piezo-1 protein actually internalize much fewer red blood cells compared to the wild-type macrophage, suggesting that piezo-1 is required in macrophage for normal and efficient phagocytosis. So all our molecular evidence propose a new piezo-1 dependent pathway for our iron overload. So in this model, we think that the mechanically activated piezo-1 protein on macrophage translate more calcium ions into the macrophage, which actually elevates the active REC1 protein levels, and then it enhances the phagocytosis of the macrophage. And this overactive recycling of phagocytosis, overactive recycling of red blood cells leads to stressful erythropoiesis and the release of erythroferron, and which inhibit the production of hepcidin. And the end result is to have excessive accumulation of iron in the blood and in the tissues. So as I mentioned, one exciting area of what we want to get into is human genetics, of asking how the E756 delete gain of function PS1 allele affect the iron metabolism in African American individuals. So to do this, I collected more than 300 serum samples from self-identified healthy African American blood donors. And in this cohort, the majorities are male, and they age from 18 to 76 years old. And from this chart, you can see the distribution of the genotypes. As expected, almost 1/3 of this cohort have the gain of function allele, and we even have 8 homozygous individuals. And first, I found that the E756 delete gain of function allele is significantly associated with higher blood iron measurement for transferrin and the ferritin value. And we noticed that this only happens on individuals over 40 years old, but not on the younger age. So this is consistent with the age-dependent effect of PS1 function in the iron metabolism in our animal models. And also we observed that in the individuals who carry the gain of function PS1 allele, there's an increase of blood iron measurement with age, but this effect is not observed in the wild-type carriers, the wild-type non-carriers.
We have a question in the chat. Tom, do you want to go ahead and ask the question?
Sure. Shang, do you think that the way the macrophage ingestion of red cell works is that the macrophages actually test the rigidity or stiffness of the red cells before eating them? Because there's some preference for they have a preference for eating older cells.
Yes.
Stiffer.
Mm-hmm. Yes.
Is that what piezo is doing there? Is it testing the stiffness of the red cells?
Yeah, that's very interesting question. I think it is very possible because it's already found that the PS1 in other systems that can detect the stiffness of some other substrates. So maybe it's also because it can detect the stiffness of the red blood cells and also other cell types in terms of the phagocytosis. So I think this is an interesting hypothesis to explore in future.
Yes, and
yeah,
would explain when it is hyperactive, the cells that are not so stiff also get eaten.
Yes. So that's why we think the overactive PS1 actually have ectopic, mechanosensing, function that also detects something that usually not be detected by the PS1 function. So that can explain how that links the stiffness of the red blood cells.
Thank you.
Yeah,
that's kind of also going back to like the half-life question, right? Because if the red blood cells themselves weren't PS1 gain of function mutants, you're basically engulfing and recycling more red cells as though they were senescent, even though they aren't. So what happens if you then induce more recycling, like with Ph.D. or have you tried anything like that? What happens?
Yeah, actually, we haven't tried that, but I think this is a a simple experiment that we can do. So yeah, we haven't.
One more question in the chat. Thomas, do you want to go ahead? Thomas Coates.
Do you know if the piezo1 can conduct ferrous iron? I asked this because of the heart question. Is this possibly a mechanism? The heart's beating all the time. The counterreceptors are probably going crazy there.
Yeah, so this is a wonderful question. Actually, piezo1 channel is non-selective chitin channel, so it can transduce all the positively charged ion. So, but we haven't tested whether it is permeable to the iron. I assume iron maybe is larger than the calcium, or I'm not familiar, but it is a it's a possible hypothesis to test.
Can you just compete with other metals? So the calcium, if you put higher levels of the metals, other metals in there when you're measuring calcium flux, does it change?
Yeah. In vitro, actually, that is the case because it's non-selective. Channel. Yeah. Okay. So and also
One
in
more
the
question. Sorry,
okay,
this is fun.
sure, no problem. Yeah,
Go ahead, Nick.
Yeah, it is. It is.
Nick Bestman.
Sorry about that. Yeah, I'm
Mhm.
just curious. So have you looked at phagocytosis of cells other than red blood cells, like apoptotic cells or bacteria? Is
Yeah.
this specific to red blood cells or?
Yeah, that's an interesting question. Actually, yeah, actually, in our experiment, we also use, you use the beads or the substrates of the not substrates, the plasma memory parts of the yeast or bacteria. And also that is the case for those, phagocytosis substrates. So I would say it's more like the general rather than specific to red blood cell in
Okay.
terms of phagocytosis. Yeah, that's
So
an
I guess-
interesting question.
So
Yeah.
in that sense, does that argue against it being a defect in detecting the stiffness of senescent red blood cells and a more general macrophage phagocytosis effect?
But I think it can still actually detect the stiffness in vivo, right?
Okay.
I mean, in vitro at this level, what you said may be, true. But
Sure.
in vivo actually is much harder to study. But I
Sure.
think it can still happen. Yeah.
Okay, sure. Very interesting. Yeah.
Thank you.
Thanks.
Yeah. And then if you remember, in the animal models, we determined that the reduction level of hepcidin is the cause for the iron overload. And in the human cohorts, I also showed that the hepcidin levels actually is significantly decreased in the individuals who carry this gain of function PS1 allele in the genomes, which is consistent with our animal models. And eventually, we want to start asking the potentially clinical relevance of our discovery. So whether the E756 delete is associated with clinically high iron measurement in the blood. So as I mentioned, that both transferrin saturation and ferritin values are commonly used in clinics to definitively to help diagnose the iron disorders. So for example, individuals with higher transferrin saturation or the ferritin values are recommended to do further tests such as MRI to be definitively diagnosed iron disorders. And then I applied these similar criteria used in clinics to our cohort. And then I found a very intriguing pattern. So in this panel on the very left, this pie chart has all the individuals in our cohort, and as you can see, 1/3 actually carry the gain of function allele. However, when I looked at the individuals in our cohort who have either high transferrin saturation or high ferritin value, the percentage of this gain of function allele in this cohort is way more than 1/3. And even more amazingly, in our cohort, we identified 10 individuals who have both high transferrin saturation and high ferritin values. And all of these 10 individuals carry the E756 delete gain of function allele. So of course, it is super important to notice that all those individuals in the cohort are self-report healthy individuals. So I think it would be necessary and interesting to monitor these individuals for further development of potentially severe iron problems later on in their life. And to summarize this part, actually, I started with an unexpected but interesting phenotype in the xerocytosis patients. And then I use tissue-specific animal models to learn something about mechanotransduction dependent on PS1 in macrophages and iron metabolisms. And eventually, I showed the potential relevance of this gain of function PS1 to the larger African American populations. Hopefully, this highlights the importance and the power of combining human genetics and mouse genetics. And also, interestingly, I would think about the impact of this very common gain of function allele in African population, because this is very similar again to the malaria story, right? So where we observe that this gain of function, the gain of function PS1 mutation offer resistance to malaria, but as a trade-off, these individuals actually develop higher iron content in the blood. And one more important question is, are there more effects to be explored in these individuals who carry this common gain of function allele?
One more question in the chat.
Yeah.
Go ahead and ask that, Thomas.
Oh, it's you said more effects to be explored. This mutation is very high in African Americans. That's very, very interesting. The fact that it dehydrates, this should make patients with sickle cell disease have much worse disease. I'm wondering if there's any way you can look at that.
Yeah, that's an interesting question. Actually, so there has been independent report suggesting that there is genetic interaction between this allele and the sickle cell allele. So, but I think it's a with a more with larger cohort, I think we can definitively study this. So that is very interesting perspective.
Thanks.
Yeah. So here are some future directions. So as I suggested that we explore the downstream signaling of how piezo-1 actually causes the enhanced phagocytosis, but actually the exact mechanism at the bottom of this, it's still not quite clear. I think by using some high resolution imaging approach, we can actually mechanistically study how the sensation links the mechanical force to the plus memory dynamics. And then another interesting thing to ask is whether the macrophage specific loss of function piezo-1 might develop the iron deficiency during aging, which is the opposite phenotype of iron overload in the gain of function animals. And then, as I mentioned, it is important to monitor, those African American individuals who carry this allele for further potential development of iron problems, including the overload. And then, are there other disorders in these individuals, and whether those potential effects due to iron overload or due to something some independent mechanism dependent on piezo-1. So, and also we can keep exploring other mechanical, medical conditions in the rare xerocytosis patients to explore more research directions of how piezo-1 are important in mechanotransduction for human biology. So finally, I want to actually thank everyone in Patapoutian's lab, both current and the former group members. So I highlight the names of individuals who made contribution to the stories I just told you. And also we have very nice collaborators who contributed to the experiment I just explained. And a special acknowledgement goes to Thomas Gans, who made very critical suggestion to our manuscript for the iron overload story. And here is the funding agency to support the work. So thank you very much. And thank you for all the questions I got so far. So I'm happy to answer more questions if you have. Thank you.
Thanks. Carolyn, do you want to unmute yourself and ask your question?
Sure. I was just curious as to whether you had encountered, since this is a fairly prevalent mutation in some populations, compound heterozygotes or compound mutations with the HFE gene, the hereditary hemochromatosis gene. I mean, I know these are traditionally found in two different populations, but it would seem like the prevalence would have had you encounter that. Are you aware?
Yes, actually, the mutation you mentioned, HFE, I think is more prevalent in the Caucasian population, but it's actually fairly rare in the African population. But you are right, with the more numbers in our cohort of African individuals, we may be able to study the interaction between these alleles. But actually, another way to study it is to explore other genetic consortiums. For example, so far we have been exploring the UK Biobank, which have 500,000 of cohesion, individuals with all the, with the whole exome sequencing available. So in that consortium, we identified the novel gain of function PSO1 mutation in the cohesion families. So there will be an opportunity to study, how the gain of function mutation in PSO1 interact with other mutations in causing the errant problems.
Shang, this is Tom Gantz. I have a question.
Yes.
So there's been a challenge to the model of red cell processing in the spleen, where it's not the macrophages that eat the red cells directly, but there is a shear in a kind of a reticular network of the spleen. There is a shear test. The cells lyse, and then the ghosts are eaten by macrophages, and the hemoglobin is recycled by macrophages as well. So there's kind of a shear test first, and the cells that get sheared get destroyed and then eaten. How would you reconcile that model? Is there a way of reconciling this alternative model that was proposed by Robin van Bruggen in Amsterdam and his group at Shang? Is there a way of reconciling it with your data if that turns out to be true?
Yeah, actually, we noticed some other alternative model for this, but so far we haven't been able to actually test it in vivo. But I think what we can start is maybe start with some in vitro experiment using the red blood cell ghost,
yes.
To test whether the macrophage actually function from there, and then we can get some hints. And we can decide whether to go to the in vivo. But so far, I don't think our current model actually can distinguish between that two.
Okay.
That's my thinking. Thank you.
Yeah. Can I ask you something?
Yeah.
So did you have a look whether the expression of piezo changes also contemporarily with the phagocytosis activity, also some cytokine expression, or did you look at that?
So you ask the expression.
Yes.
The expression of piezo in the macrophage.
Yes.
So are you asking whether there's a difference between the wild type and gain of function?
No, I'm asking if changing the expression of piezo, you also change the action of cytokines in macrophages together with the phagocytosis.
I see. Yeah, I think we measure the cytokines in the spleen, but we didn't actually in the spleen of the macrophage specific gain of function animal, but we didn't detect significant difference.
And after doing phagocytosis assays?
No, actually, we didn't do that after phagocytosis.
Okay, I think that would be interesting.
I see. Yeah, I see.
Other questions? It's been a great discussion, really fun.
Yeah, I really enjoyed that.
Okay, if there are no other questions, it's nice to see everyone, and I reach out to Shang if you have questions or ideas that you could help assist in the science and going forward. This has been really fun. Thanks for everybody's attendance, and Shang, thank you again.
Yeah, thank you very much. Yeah, I really enjoyed that. Thank you for the opportunity. Yeah, have a nice day.
Bye.
Yeah, see you next time. Yeah,
Bye.
thank you.